All right, ready, set, go. Let's take a look at neurons again, their structure and function, and at least where this is a little elaboration on what we did the other day. So this part here is the same. So these are just different ways to classify neurons. We can look at them by their structural differences. We can look at them at what they do functionally. And so we went through this unipolar, bipolar, and multipolar neurons as examples. We also said we can look at them in terms of what they do. So we have sensory neurons. We have relay neurons that are sending information certain distances, very often in the cerebral cortex. These sensory neurons would be ones that we have in the peripheral nervous system because they're going to pick up sensory information such as touch or heat or pain or light. And then we have the motor neuron right here, which is one that is going to be coming from the nervous system, and we'll get into that terminology too. So exiting the nervous system, this will be an efferent type of neuron, where this would be an afferent neuron. So let's look at that here right now. So we can classify them by their direction of the flow of that information. So something that is efferent is neurons which exit the central nervous system and then go toward things like muscles and glands and other things that they're going to effect. So it's an efferent neuron. An affector neuron is one that does the other way around. So it's something that's picking up sensory information and sending it into the central nervous system so it can be processed. We can look at neurotransmitters and we can classify neurons by the particular neurotransmitters that they would release. They could release excitatory neurotransmitters, which are ones that are going to cause the signal to move on, to continue, and that signal we call an action potential. One example of an excitatory neurotransmitter is glutamate. An inhibitory neurotransmitter that can be released would be something like GABA, and that's going to inhibit activity, it'll stop activity. And modulatory is going to do something different. So it's going to modify, it could amplify, it could tone it down a little bit, so it has this ability to change it slightly. And serotonin was an example. Then we kind of looked at it in a real broad sense. We said, here's a neuron without any of the frills. I mean, this is an unmyelinated neuron, and we know that neurons pick up information on this side and they transmit it down the axon. So this side over here, where the dendrites and the soma are, the input zone, there's the axon hillock that takes all the information that comes in and decides what to do with it. That's the integration zone. We have the axon, which is our conduction zone. And then we have the axon terminal, which is the end here, which is the output zone. And we're going to get into specifics on all of this. So then we said, okay, great. Now let's talk about how this neuron receives information or signals, and then what does it do with them. So a name for a signaling molecule is a ligand. And ligands can be chemicals. They can be some other types of ligands that can activate a receptor that would be found very often on these dendritic branches or spines. Now, a receptor is a protein. And so what I did here is in this little blue box, I tried to elaborate just a little bit on it here. And so the brown that you see there is representing those little squares or receptors. And here you can see really much more what it would really be like. So they're proteins. And so you can kind of see this long chain here that's all sort of wound up here. So this would be an example of a type of receptor. Notice that this is the outside. So this was just a little piece of that, right? So we're just looking on the outside of the neuron, the extracellular side. And here's the intracellular side. And then this receptor, which is spanning this membrane. So there's our phospholipid bilayer, right? Like we've learned that all cells have. And we can see this protein or this protein receptor, which is sort of winding its way down here, it's actually integrated into that membrane. So some of the ligands we mentioned already because we looked at neurotransmitters. So at least we understand that these things exist: glutamate, GABA, serotonin, all being chemicals that could serve as a ligand. Also things like heat and touch and light. So there's other stimuli that can activate receptors. So, but neurons they have kind of they're either off or they're on. So a neuron at rest, well, I say that, I have to already qualify it. A neuron at rest is not sitting there doing nothing. It's more, however, like your car idling a bit. So it's still doing all the things it needs to do as a cell to maintain itself. And the nucleus is, in here we'd have DNA. We have all the other things in the soma that you'd expect and to see in a cell. However, a resting neuron has been measured to have a difference in the potential, which is we're looking at the positive and negative charges across the membrane. So all around this is the cell membrane. All of this is a phospholipid bilayer. So if we stuck a probe outside the membrane and we stuck a probe inside the membrane to measure the voltage, the electrical difference between the inside and the outside, we would see a difference based on different concentrations of positive and negative ions that would be on one side of the membrane or the other. So in general, the outside or the extracellular area in general has positive charges, and there's a high concentration of sodium outside. So here's the sodium ions. Notice the plus charge there. And then inside, there are positive charges and negative charges. And the negative charges are the large negatively charged proteins, which are pretty much stuck inside. They can't go in and out of channels. They're large molecules. So the inside is negative relative to the outside. And that measurement that we're going to use as our sort of rule of thumb for a resting neuron is negative 70 millivolts with a little m and a big V. So that's the difference in membrane potential or electrical potential, voltage potential. Notice I'm using a lot of the same, I'm saying potential with all of them, but we're looking at that difference in the charges across that membrane. Now, some of the ions that we see around cells and around neurons or within them are some of them here. So we're going to see those ions or those charged particles within those fluids that are within the cell and outside of the cell. So the extracellular fluids and the intracellular fluids, such as you know as cytoplasm or cytosol. Here are some common ones. Here's the ones we're really going to talk about mostly. We said already these large negatively charged proteins. We have potassium in large concentration inside the cell. We have sodium in large concentration outside. We have chloride outside, and we have calcium. So we'll see what their roles are as we go forward. So when we're looking at this, I tried to set the scene here. This is an unmyelinated neuron, so that we could just see the things we're really concerned with at the moment. We know that the myelin sheath, many neurons are myelinated, not all, but many, because those neurons are able to conduct the information, or this thing we're going to call an action potential, much more rapidly. But for now, what we see here are these little spots out here representing, I tried to color code, but sodium is all the orange dots out here. We have potassium, which are some purple ones. They're in a little bit in here. You can't see them too well. There's a calcium, the bigger kind of lavender looking one. And then we have these large negatively charged proteins, which are the red blobs. And so this is basically what a cell would be at rest. So it's not doing anything. It's just the input zone is waiting for information to come in. So here we have the neuron at negative 70 millivolts, resting potential. Sodium ions, high concentration inside. Potassium ions, Sodium ions, high concentration outside, extracellularly. Potassium, high concentration inside, on the intracellular side. And then the large negatively charged proteins are also inside. We know that ligands are chemical messages or some other type of message that can activate some receptors. And we also learned the other day that the receptors that are here, the brown ones that we pointed to first here on the dendrites, those are called ligand-gated ion channels because they, when they are activated by a ligand, will open a channel and allow some ion to flow into the inside of a neuron. How they do that is when the ligand, like this, which is represented with this blue spot here, lands on that protein receptor, it's going to change its shape. It's going to cause a conformational change. And that change of the shape of protein will open up the channel that is associated with that receptor. So as many of you are sick of hearing from me, shape is destiny in biology. So when we change the shape, we've allowed something to happen. Once that happens, we get some ion that flows into the cell body and here in the soma. Then that ligand, it leaves, it doesn't stay on the receptor, and it gets usually cleaned up by our friends, the microglia, or some other way of removing the ligand from that area. And we'll see some examples of that too coming up. So ligand leaves, okay? Number 3, the ion then flows in, which I already said. And if we're talking about things like sodium, which we started with, we're going to get positive charges then that would come in. Now, if a positive comes in, it's going to make the inside of the cell more positive, aka less negative. So keep that in mind. When those positive charges come in, that's going to change the inside of the cell. It's going to change the membrane potential. And the axon hillock is going to determine if it's significant enough to do something. And if that voltage difference on the inside becomes positive enough that it reaches a mark we call threshold, then the axon hillock is going to open a gate, but this time it's going to be a voltage gate, a voltage-gated channel, and it will do that in response to once it's added up all these, and if these plus charges have reached a threshold, in other words, the cell has become positive enough on the interior right here, right now, then this V gate that you see here, or voltage gate, this very first one, is going to open, and that's the axon hillock's job. Now, we're going to talk about then where it goes down this axon. So it's only this first gate initially that's going to open. Sodium comes in here, though, and it comes in a large quantity, and that causes this part of the membrane on this inside of the axon to also become negative, or positive, or more positive. So it's going to trigger the next gate opening, etc., etc. So the current actually is flowing down this axon, and what we have is the voltage change is going to happen in sequence like dominoes all the way down the axon. This is one way to look at it. We're going to talk about this in class. So here's the membrane potential. Here's negative 70, so this would be the resting potential. This would be what the neuron was at rest. And now we have sodium coming in, and notice that this in this case, what we see is a little blip here, a little excitatory activity, but we don't see that we haven't reached threshold yet. So we're going to talk about that in class tomorrow, but I thought this would be a good way to summarize this, and then we'll take it from there. So stay tuned.